Hello students, welcome to Digital Learning DCE. Myself Dr. Manjula S, working as Assistant Professor of Botany. In this session, let us study about a brief account of immune systems, application of immune techniques in agriculture and industry, monoclonal antibodies production, and ELISA techniques. To begin with the introduction, the resistance of the host towards a pathogenic microbe and its toxin is known as immunity. Immunity is of two types, natural and acquired. Natural immunity is otherwise known as heritable since it is present in an individual during their birth time. They are broad-based, hence they are also non-specific in nature, whereas acquired immunity are acquired during the life of an individual which are specific against a particular pathogen. Immunology is the branch of biology which generally deals with the study of defense mechanisms of organism against the infectious agents, which also analyzes the biochemical or molecular basis of immunity. In the immunology, we came across certain important concepts like antigen and antibodies. Antigen is a substance. When introduced into a host, it induces or provokes the immune response, which leads to acquired immunity and results in the formation of antibodies. Whereas the antibodies are specific substances produced as a result of antigens. They are globulin protein, hence they are also called as immunoglobulins, which are of 5 types like IgG, IgM, IgA, IgD and IgE. Each antibody is specific in its function, where IgG is the most common type which provide immunity to newborns. Coming to the types of immune system, the system which produces body's defense against disease-causing organisms, malfunctioning cells and also against the foreign particles. It has 3 major functions like protection from disease-causing invaders, removal of dead and damaged tissues and cells from the body, and it also involved in recognition and removal of abnormal cells from the body. The immune system is of 2 types, humoral immunity and cell-mediated immunity or immune system, where the humoral immunity involved in the production of antibodies, whereas the cell-mediated immunity directly act against the particular pathogen. The cells involved in immunity includes macrophages, B cells and T cells. Coming to the monoclonal antibodies and its production, an antibody which is produced from a single clone or a single cell line is called as monoclonal antibody. It consists of identical antibody molecules which targeting a single epitope by having same antigen binding site. The monoclonal antibodies are produced by a special technique called hybridoma technique, which involves the fusion of immunized animal B lymphocyte cell with a myeloma cell, which results in the formation of hybrid cell called hybridoma, which later cultured to get monoclonal antibodies. In this diagram, we can observe the steps involved in monoclonal antibody production, where the mice experimental animal which is injected with the desired antigens. As a result of that, the spleen cells from the blood of an injected experimental animal mouse is extracted, and the spleen cells are allowed to fuse with the myeloma cells. When these 2 cells are fused together, a hybrid cell is going to be formed called hybridoma. This hybridoma are cultured in HAT medium, select for positive cells, and from this selected cells, the monoclonal antibodies have been harvested. The monoclonal antibodies have so many applications, like they are used to detect some plant viral diseases. They also used in identification of blood groups. Monoclonal antibodies are used for diagnosing and monitoring effectiveness of cancer therapy. They are also used in high-level purification of medically important substances. Coming to the next immunotechnique, that is enzyme-linked immunosorbent assay, which is abbreviated as ELISA. ELISA is a common laboratory serological technique which is used to measure the concentration of antigens or antibodies in a solution. Here, the antigen antibody of interest is absorbed onto a plastic surface that is termed as a sorbent, where the antigen is recognized by a specific antibody that is otherwise known as immuno, and this antibody is recognized by a second antibody which has enzyme attached, hence enzyme is linked, where the substrate react with the enzyme to produce a color product in this assay, hence the name enzyme-linked immunosorbent assay. So in this diagram we can see a ELISA plate which is used to detect antigen antibody reactions after the formation of color change in this plate. Then move on to the application of immunotechnics in the field of agriculture and industry. ELISA is widely used in agriculture to detect plant viral diseases like potato virus such as PVX, PVY and PLRV. ELISA can used to detect plant viruses even in the absence of disease symptoms, hence it is very specific technique. Monoclonal antibodies are used to detect several bacterial diseases which are also have the therapeutic applications which are directly used in destroying disease causing organisms. Monoclonal antibodies used in food industry to detect foodborne pathogen and also mycotoxin testing. ELISA can also be used to test specific allergens. Qualitative detection of food allergens can be done by using dot immunoblotting or STS page, but rocket immune electrophoresis and ELISA are used for quantification of hidden food allergens. In food industry, ELISA technique is used for detection of adulterations and also mycotoxins present in the food. Coming to the session summary, as I told, the resistance of the host towards pathogenic microbe and its toxin is known as immunity, which is of two types, natural and acquired. The natural immunity is non-specific in nature, whereas the acquired immunity is specific against a particular pathogen. The study of immunity to infectious disease or study of structure and function of an immune system is called as the immunology, where the immune system produces body's defense against disease-causing organisms or malfunctioning cells and also against the foreign particles. The monoclonal antibodies are the antibodies which are produced by the single cell line, hence they are identical antibody molecules targeting a single epitope by having a same antigen binding site produced by a special technique called hybridoma technique. ELISA is a common immunological technique which is used to test used in laboratories to study antigen antibody reactions, which is widely used to detect some important bacterial and viral diseases, whereas the monoclonal antibodies are used to detect foodborne pathogens and mycotoxins. So with this, I am going to conclude this session. For further details about the current topic, you can refer the following references. Thank you.